Hey all, welcome back. I've got my coffee and we're good to go. In this lecture, we're going to talk about mutations. Particularly, we're going to talk about point mutations and frameshift mutations and the effects that they have on DNA sequence, mRNA sequence, and the sequence of a protein. Let's first talk about how mutations originate. Mutations are changes to the sequence of a DNA, and they may occur spontaneously, or mutation rate may be increased with the use of mutagens. Let's first talk about spontaneous mutations. These spontaneous mutations occur during S phase of eukaryotic cell cycle when DNA is being replicated by DNA polymerase. In bacteria, DNA polymerase makes one mistake or one mutation every 100 million to 1 billion nucleotides. Put in scientific notation, this means that there's one mutation every 10 to the 8th to 10 to the nucleotides that is replicated. Let's put this into perspective of the human genome. The human genome contains somewhere around 6 times 10 to the 9th bases. Given the lower of the two mutation frequencies, which is 1 in 10 to the 9th, we could assume that there are about 6 mutations that occur every S phase within a human genome. Now, the good thing for us is that many of these mutations are repaired by additional repair mechanisms that we're not going to discuss in this video. Mutagens are chemicals or other substances that can increase the rate of DNA mutation. UV light is such a mutagen. In this example here, what we're looking at is a strand of DNA. And when UV light from, for example, sunlight comes and hits this strand of DNA, it causes the DNA to change shape. In fact, what it does is it creates a covalent bond between adjacent thymines. And this is known as a thymine dimer. You'll notice here that this thymine dimer causes a little kink or a little bend in the sugar phosphate backbone of the DNA. This kink in the DNA can affect both replication and transcription and increase the likelihood that mutations will occur. Luckily for us, we have enzymes that recognize these lesions of thymine dimers and can help to repair them. Additional examples of mutagens that can increase the rate of mutation include chemicals like ethidium bromide, sodium azide, and other rays such as X-rays and irradiation. There are two major types of single nucleotide or single base mutations, and we call these point mutations and frameshift mutations. We're first going to talk about 3 types of point mutations. You'll notice in the top left-hand corner here we have our original DNA sequence. We're just going to define it as original for the sake of this scenario. And remember that template DNA and mRNA are anti-parallel and complementary to one another. And so the DNA sequence produces the mRNA sequence shown below it. And if we divide that mRNA into 3-letter codons, that will help us to determine the amino acid sequence for this protein. So for example, AUG codes for methionine, CGG codes for arginine, and so on and so on. So let's take a look at the first type of point mutation. You'll notice here in our new DNA sequence that where we originally had a C, we now have a T. This affects the mRNA sequence, and now in that second codon, instead of CGG, we have CGA. However, this still codes for the same amino acid. Both CGG and CGA code for arginine. This type of mutation, where a new codon is produced but it codes for the same amino acid, is known as a silent mutation. In other words, it has no effect on the amino acid or the protein structure. The next type of mutation we can see in the bottom left-hand corner. In this example here, again, where we originally had a C, we now have an A. That's going to affect the mRNA sequence and now changes that second codon from CGG to CUG. CUG codes for the amino acid leucine. And so in cases like this one here, where a mutation in the DNA creates a codon for a new amino acid, we call these missense mutations. Finally, in the bottom right-hand corner, we have our third type of point mutation. In this example here, you can see that before, in the original DNA sequence, we had a G, and now we have a T. This T, when turned into mRNA, or when transcribed into mRNA, would code for an A. And you can see that this has disrupted that third codon. Where before the third codon was UGC, coding for cis, this codon now reads UGA, which is one of the three major stop codons. And so this type of mutation where a stop codon is generated is known as a nonsense mutation. The name comes from the fact that sometimes these stop codons are also known as nonsense codons because they don't code for an amino acid. Now that we've taken a look at 3 different types of point mutations, I'd like you to practice with this template DNA shown here. Go ahead and pause your video to create silence, missense, and nonsense mutations within this template DNA sequence. After you've practiced with it, go ahead and move on to the next slide. Frameshift mutations are created in which one or more nucleotides have either been removed or added to a DNA sequence. Again, we're going to start out with our original sequence here, break our mRNA into codons, and we can find we're working with the same sequence as we did in the last example. Let's first take a look at a deletion. A deletion is a case in which a nucleotide has been removed, and this is going to disrupt our codon sequence. You'll notice here that the 4th nucleotide, which was originally a G, has been removed. This causes a frame shift, or a shift in the reading frame of the mRNA sequence. So before, where our second codon read CGG, our new second one reads GGU. Where our third codon in the original sequence read UGC, it now reads GCA in this mutated sequence. In cases like this, deletions are going to affect every single codon downstream of where the mutation has occurred. Similarly, insertions can have a very similar effect. In this case here, you can see that a G has been added. This is going to shift the reading frame and again change codons after that first one. And so now you can see here that our second, third, fourth, and any additional codon would be different than what we had in the original sequence. Interestingly enough, deletions and insertions have their smallest effect when 3 nucleotides are either deleted or inserted at a time. For example, in the bottom right-hand corner, I'm showing you a case in which the 3 nucleotides GCC have been deleted. You'll recall that these coded for the mRNA CGG, which then coded for the amino acid arginine. And you can see by these 3 nucleotides being deleted, we no longer have the arginine, but we've maintained the reading frame for the remainder of the mRNA sequence. In other words, we still have the same codons and the same amino acids, just with the exception of that arginine. In summary, base pair substitutions are typically single nucleotide changes that occur either spontaneously or as a result of mutagens. We can divide these types of mutations into point mutations or frameshift mutations. Point mutations can be either silent, they can be described as missense, or they can be described as nonsense. Whereas frameshift mutations will occur with insertions and deletions. Until next time, take care.